Okay, pwede na tayo mag-start. Feb.
Okay.
Okay.
Okay. Let me see. Let me see.
Okay, let me give some opening remarks a little bit, just because we have uh, different format today for Philippine month. But anyway, good evening, everybody in the U.S., and good morning to everyone in the Philippines. So welcome to our month listeners. We are speaker, I would like to let you know that we are holding a series of 3 seminars, which will commemorate the Filipino-American History Month celebration. So there will be 3 webinar series, the first of which will be today, and I will leave our introducers to introduce our speaker. But so you know that what this Filipino History Month is. I will introduce Dr. Mario Santo Domingo, who is our ex officio president. Immediate past president will give an introduction as to the significance of the Philippine American History Month, which is currently being celebrated all over the United States. In fact, our Filipino organization here in the U.S. has a series of activities commemorating this event. And so we have a series of 3 seminars, the first of which will be today from Dr. Vidal. The next one will be next week, will be given by Dr. Xenia Agustin from University of Illinois. And then the third one will be actually in person and also via webinar by Dr. Gobet Avincula, who will be giving it in person from the University of the Philippines in Los Baños, and also online for those who cannot be there in person. So Welcome to our celebration of the Filipino-American History Month. This is the first time that we have celebrated this. In fact, many of us here, even those of us in the US, didn't know about it until we finally started celebrating it and recognizing the significance of this month. So, to give an introduction of what the celebration is, let me give you Dr. Mario Santo Domingo, ex officio president of the PAASE and our immediate past president. Mario?
Maraming salamat, Marge. Magandang umaga po sa ating lahat, sa mga nasa Pilipinas, at magandang gabi mula dito sa Estados Unidos, mula dito sa siyudad ng Baltimore. Ako po si Mario Santo Domingo, ang dating Pangulo ng PAASE. So I was requested by President Gladys Completo to provide some context to why we are here today, what we are celebrating, and the other details in connection with this celebration. So today, actually, on October 18th, the first group of Filipino settlers arrived at Morro Bay in California. That was in 1578, if I'm not mistaken. And until today, I wonder whether all of us, or most of us, are familiar with the Filipino-American history and how many Filipinos are now in the United States. Based on the last census in 2020, there are now over 4 million Filipinos living in the United States today. So this is a huge number, and it's very significant across different areas of our lives here in the United States, and also in the Philippines, because it has a lot of implications in terms of what's happening in the Philippines as well. So the Filipino-American History Month is celebrated every October to honor the contributions of Filipino-Americans to U.S. history, culture, and society. So as I've mentioned, the first recorded arrival of Filipinos in the U.S. Dates back to 1587. So that's the correct year. And coincidentally, it's October 18th. So it's October 18th in the Philippines today. And they arrived in Morro Bay, California then. So the first set of Filipinos were called Luzones Indios. They're Filipino sailors who arrived on the Spanish galleon, the Nuestra Senora de Esperanza, and it's part of the Manila Galleon trade, a global network that connected Asia, Europe, and the Americas from 1565 to 1815. The celebration also recognizes the significant migration waves, including Filipino laborers in Hawaii and California in the early 20th century, and the impact of the Immigration and Nationality Act of 1965. So this particular act abolished quotas, established a preference system that favored immigrants with skills, family ties to U.S. citizens, or refugee status, and opened the door to non-European immigration. So you can just imagine, the impact of this act, in terms of migration of people from across the world, not just from the Western Hemisphere after 1965. So since then, of course, and even before, the Filipinos have contributed greatly to the lifeblood, of the United States, and that's including, of course, healthcare. Do you know that during COVID, 24% of the nurses who died of COVID were Filipinos? And of course, we have many other areas where Filipinos contributed very significantly, including the sciences and engineering and medicine, which are the most relevant areas we are actually concerned about tonight. So one of the results, of this migration of the Filipinos to the United States, of course, is like the gathering of a significant number of scientists who are trained in the US and also across the globe. And in 1980, a group of Filipinos organized the Filipino-American Academy of Science and Engineering, or PAASE, to promote scientific collaboration between the US and the Philippines. Its goal is to bridge the gap between Filipino and American scientists, fostering innovation and enhancing the global Filipino community's contributions to science and technology. The PAASE was founded by Dr. Severino Ko. He's like a professor of engineering at Purdue then in 1980, and then transferred here at the University of Maryland. The first ever conference was held in Manila on January 6 to 8, 1981, and the rest is history. The current president is Professor Gladys Completo from the University of the Philippines, Los Baños, and the vice president is Dr. Marge Peña from the University of South Carolina in Columbia, in South Carolina. I would be remiss if I will not mention again this very important project of PAASE, which is SciEngJ, because we are investing heavily on this. I'd like everyone to invite everyone to contribute to SciEngJ. Please visit scienj.org and see how you can submit your manuscripts, and we will very much welcome, your contributions. So that's it, folks. We would like to again thank you for being with us today. Please continue exploring the Filipino-American history and engage with the global Filipino community through events and collaborations. Maraming salamat po.
Thank you, Mario. Thank you for that very nice introduction and for giving us the history of the first Filipinos who arrived in the United States. That history has really been spread to all of us who are here. And so even our Filipino-American student associations are holding celebrations of Filipino-American History Month throughout the month of October. And in fact, our organization created one of the first documentaries about the Mangyans, who were the indigenous culture in the Philippines long before the Spaniards came to colonize the Philippines, indicating that we had our very own rich culture, our very own unique languages, and the unique ways of arts and communicating with each other. So one of these days, I would like to share that with the PAASE. It was created by one of our colleagues here in cooperation with the Mangyan Association in the Philippines that is very well protected and preserved. So without further ado, we would like to invite Dr. Billy Cabillon to introduce our speaker today. And Dr. Billy, please take over.
Thank you. Thank you, Madam Vice President, and thank you, Vice President Mario Santo Domingo. I am Silverio Caballon Jr., a graduate of the University of the Philippines College of Medicine. And my colleague this evening, who will be posing the question and answers, is Dr. Orlad Tantenko. I think he presently is in Texas. Before, in the light of the theme of Filipino-American Month, October, I would like to digress just a little bit for a short period. The introduction of this webinar alluded to Philippine Science and Engineering Journal. This week, in the light of Philippine-American friendship, I learned that the Nobel Prize winner in medicine, Dr. Rosalind Yellow, published in Acta Medica. She was the Nobel Prize winner in 1967. And lo and behold, the lowly Acta Medica Filipina published her paper, and then in 1967 won the Nobel Prize. So hopefully the Philippine Science and Engineering Journal will one day also have the writing of a Nobel Prize winner from the Philippines. So let me introduce Dr. Manuel Vidal Jr. Dr. Vidal is a newbie. He just earned his PhD degree just this year, and he did his research in Texas at the University of Texas branch in Galveston. He holds now, of course, a double degree of medicine and a PhD. He topped the chemistry licensure exam in 2000 he ranked second in the licensure exam in 2013 and graduated magna cum laude in biochemistry from LaSalle. So Dr. Vidal caught my eye because he is a prolific writer and he funded his own studies, no money coming from the government, and he was able to publish 3 really great papers. And with that background, he is now involved with COVID-19, immunotyping for COVID-19, liver research, preeclampsia, which is his topic tonight, and antenatal screening research. He presently is a faculty member of the Department of Physiology of Beda University. I would be remiss if I did not mention that he also belongs to Pi Kappa Mu fraternity. So, Dr. Vidal, sir, the floor is yours.
Hello, hello.
Okay, there we go.
Sorry. Well, hello. Can everyone see my screen?
Yes.
Right.
Oh, on the screen.
Thank you everyone for the opportunity po, especially Dr. Cabellion for especially reaching out for me to share this wonderful work that we have done in Texas. Actually, this project is a step towards a future where we are not going to have any more human subjects or animal studies for drug testing. This work actually was done in Texas, where Dr. Tentenko was my immediate senior in that lab, and I came in 2 years after. And he has done extensive work with cervical cells. But for me, when I came into the lab, my primary interest was the placenta. So let me share my screen. Wait, just hold on. There, can everyone see my slides?
Yes, yes.
Yes.
Yes, we yes.
Thanks so much. So, well, again, I'm thanking Dr. Cabellion and everyone from PAASE for giving me the opportunity to tell you about our work. This is my thesis, actually. So we are developing new alternative models for the placenta. So this paper has been published in, this work has been already published in 2 journals. So in the Royal Society of Chemistry and Biology of Reproduction. And so here, we are going to talk about pollutants. And how do they impact the placenta? So as we all know, the placenta is the barrier towards the fetus. So it's exposed to a lot of pollutants as diet and natural exposures occur in pregnant humans. So one of the pollutants that we are interested in are endocrine-disrupting compounds. So these are bisphenols and PBDs. So these are compounds from plastic products. So whenever they become exposed to the environment, they get leached out of the containers, and they become free molecules, and which they deposit into natural reservoirs in the environment, such as the water and the soil, and that leads to human intake. So not only this is an environmental problem, but it's also a human health problem, because these compounds have been associated with a lot of different effects when it comes to fetal health. Some of their effects in pregnancy would include changes in vasculature, changes in hormone production, and most especially preterm birth. So the placenta is a fetal organ, which is a barrier for xenobiotics, and it facilitates nutrient transport. Since these pollutants induce certain placental dysregulations, we want to know how do they work. Because if these plastic pollutants are being consumed in every day, then why don't we see a big spike in preterm birth rate? So we want to explore how these pollutants induce placental damage, contributing to preterm birth. So we ask this question, and we are hypothesizing that these compounds, once they get transferred across our model, it would induce a lot of damage that leads to placental function dysregulation that will lead to preterm birth. So, of course, the placenta is a fetal organ, so we don't really have any human alternative for that. So in the traditional literature, they have done 2D cultures of cells. They have taken tissues from the placenta. They have used animal models also. And some of them would even have an actual placenta that has been already delivered, and then just make it alive for a few more hours. So these are traditional models. What the lab would want to do and explore is the possibility of a newer model. This is called an organ-on-a-chip. So these, if you see this one, let me zoom this in. So that is just the size of a 5 peso coin. And that chip, we call it a chip, that chip will contain different chambers that will have different cell types from the placenta, and we connect them using channels, allowing them to function as a single unit. Imagine if you have a 5 peso coin in your hand, that's the size of, that's how we read, that's already the placenta. So in the lab, they have done a lot of different organs also, mainly focusing on reproductive and gestational organs. So first we have fetal membranes, we have the fetal maternal interface, we have the cervix, which Dr. Tentenko has already done before. So this is a model of the cervix. It's smaller than the 5 peso coin. I've seen it before. That's a cervix, and they have done a primitive model of the placenta. But what we saw is that their current model of the placenta, it's only 3 chambers, which you only have the placental cells, and here you have the fetal endothelium, the fetal vessels. And so what we were asking is that, can we improve on this model? So first, if we are going to have a proper model, then we need the proper cells, and we need to have the proper chip design. So this is a schematic of the placenta. So here is the maternal side. Here is the fetal side. And this is what connects them, the vessel. And the placenta here is what comes out from the fetus. So first, we need the proper cells, because traditionally, if you observe here, all of these ones are cancer cells that are being used as models for the placenta. So what we thought of is just take an actual placenta and maybe immortalize them, make them alive for a few more weeks, and then we can use them for our experiments. So what we did, we took some placenta from term patients, let them deliver, we get the placenta, we take that placenta in a box, and then we digest them with a lot of different enzymes, get the actual cells in the end. So we have isolated an actual placenta and some mesenchymal cells, some stromal cells. Here, what we did next was to immortalize these cells in order for them to survive a few more rounds of experimentation. So as you can see here, they don't really look any different from the main human cell that we have isolated. And we also have shown that these cells, once you give them exposures here with cigarette smoke extract and a bacterial antigen, they respond similarly to human cells. And when we check their images, we see that they also light up similarly as with the normal human cells. After that, we tried to differentiate them into another cell type. We call that the syncytial trophoblast. So in the placenta, you have two main types of cells. You have the main trophoblast, which is the supporting cell for the fetus, and we have syncytial trophoblasts, which secrete all those majority of the hormones that are being expressed in pregnancy. We treated them, transformed them into these kinds of cells, and we see here that when we compare them against the standard cancer cell line, they're not too different at all. They also express the same markers, they look the same, similar to the commercial cell lines. Now, to prove that our cells were more superior than the traditional cancer cells, what we did was we took the entire RNA from them using our cells here, and we compared the transcription machinery of them versus the commercial cell lines, the commercial cancer cell line, and one of our previous trophoblast cell line. If you see here, group 1 here is comprised of our cells. Group 2 here comprises the BO cells, or the cancer cell lines that have been traditionally used in the literature. And group 3 is the other trophoblast cell line that we have used also as a reference. And we see that both of ours are way, way different when it comes to the gene expression compared to the traditional cancer cell lines. So when we treat them also with a lot of different markers, we see that they are able to show, they are able to differentiate into syncytial trophoblasts, which are the other cell types. So in summary, we were able to immortalize primary cells, make them fully functional over a few more weeks, and we can differentiate them to the placental cell that we see in actual human samples. And our cells are transcriptionally and functionally distinct from the other trophoblast cell line and from the traditional cancer cell line. So that's just part one of our study, because we needed, again, the proper cells. Now, these are not available, but after our experiments, they are now available in-house. Now we need the proper chip design, because it should contain all of these layers. So what we did under my professor is that we created a model that would incorporate different chambers, here shown in different colors, add the cells into those chambers, and that's already the chip. So that mimics also the blood flow from the mother here, from the maternal circulation, going down to the maternal tissue, and going down to the placenta, and finally ending up in the fetal vessels. So in this chip, not only we were able to include all of the basic cell types in the placenta, we were also able to show that these have the same flow from the mother to the fetus. So what we did to validate them, we added the chip, we added the cells into the chip, and then we checked their morphology, we checked their cell-specific markers, we checked for collagen production, and finally, we checked hormone production. So we were able to see that they maintain their morphology, they express cell-specific markers expected for them, and they are able to produce hormones, similar to a fully functional placenta. And after 72 hours, we see not much cytotoxicity, meaning that we can explore and experiment with them for 3, maybe extend them to 5 more days. So we also showed that our cells, our chip, are able to produce that flow. We introduced a compound, bisphenol. We introduced this compound into the maternal circulation in that chamber, and we see that after 3 days, we see a gradient forming down, meaning that anything that we put in that chip, starting from the maternal circulation, eventually finds its way to the fetal circulation. So that's across the 3 days. So in summary, we are able to show that our chip is a stable model that allows anatomical and functional representation of the placenta, and we can use this for drug screening, drug propagation studies, and function assessment of the placenta. So we have the proper cells, and now we have the proper chip design. So now it's time to do the actual experiment. So this process maybe took a little bit over a year, maybe 1.25 of a year, because the cell lines, we did this for about 9 to 12 months. We did the chip here for 12 to 15 months. And then we did the final experiments in the last 3 months of my stay in the US. So now we have a better model for the placenta. So first, as a proof of concept, we introduced cigarette smoke extract. So cigarette smoke, of course, we all know would generate a lot of radicals, would induce oxidative damage, and will induce cell death. So we see that when we treat this, the chip with cigarette smoke extract, it suppresses antioxidant capacity, it induces cell death phenotypes, and it creates a localized inflammatory environment. What is interesting here is that some of the cells would get inflamed, but most of the cells would compensate for that by creating an anti-inflammatory environment, meaning that the placenta rescues itself. So in summary, oxidative stress from cigarette smoke extract can induce redox balance dysregulation that leads to cell death and localized inflammation. So that's a positive control. We need something to compare our compounds against that control. So we now went to the actual compound. So how do bisphenols affect the placenta? So in a similar fashion, bisphenols suppress antioxidant capacity. It may activate survival in fetal vessels. And BPA, bisphenol A, causes endocrine disruptions. BPA also induces an apparent survival phenotype. And similar to cigarette smoke extract, it creates a localized environment. So in summary, we see that there is a global reduction of antioxidant capacity, similar to cigarette smoke extract. It produces cell-specific endocrine disruption, meaning it disrupts hormone production in some of the cells, and it creates a localized environment. Now, in the literature, they have said that, oh, as an alternative to bisphenol A, we can make bisphenol S. That's probably a safer alternative compared to BPA, because if you buy products in the US, you're going to see that plastic products are always branded as BPA-free, right? Because mainly they are scared of the health effects of bisphenol A. So to replace that, BPA-free products are replaced with an analog called bisphenol S. So we want to see how does that affect the placenta. Similarly, BPS suppresses antioxidant capacity in non-vessels. It induces fetal vessel imbalance, and it also causes endocrine disruption. It also induces cell death, but there are some pro-survival phenotypes. And finally, it also causes localized inflammation. So as you see, the trend with the safer alternative BPS is that it doesn't really produce that much of the damage, but it induces localized damage as well. So it induces fetal vessel oxidative imbalance. It produces endocrine disruptions, and it creates also a localized inflammatory environment. Now, the final compound that we want to test is the pollutant called PBDE. Now, PBDE, it's a flame retardant. So meaning that it's usually applied to furniture so that they don't catch fire that easily. So these PBDEs also go into the environment. Whenever we throw out furnitures, they leach out of the environment, they get deposited, and we take them in once we get soil products from the soil. So in a similar fashion, PBDE suppresses antioxidant capacity, but not to all of the cells. PBDE activates multiple signaling pathways, and PBDE causes minimal endocrine disruptions. So PBDE also induces cell death, but with concomitant pro-survival phenotypes also. And PBDE causes inflammation only in a single tissue. So in summary, it induces antioxidant decrease to a lesser extent than bisphenols. It also activates signaling pathways in different placental cells, creates endocrine disruptions, and creates a localized inflammatory environment. So if we compare all of them against the positive control, smoke extract, we see that the cigarette smoke is of course, the most harmful one, it globally decreases antioxidant capacity and induces cell death. But if you look into the other compounds, they don't really do that much of a damage, right? So they also decrease antioxidant capacity, but they don't really cause a lot of cell death. And most of that is because of the other cells in the placenta trying to rescue the inflamed cells. So the main question here is that we are showing all of these specific dysregulation, cell death, inflammation, but how do they overall affect the movement of immune cells? What we did was we added immune cells into the chip. We tracked their movement across all different cell line, across all the different chambers. So starting from here, from the middle chamber, they should move, going to the maternal side and going to the fetal side. And we see that here, one of the tissues, most of the tissues would cause a movement of immune cells, stopping short in these three chambers. Whereas if you see cigarette smoke extract, it causes immune cells to move across all chambers, meaning that it induces more inflammation, more movement of immune cells. But the other compounds, they only cause minimal movements of immune cells. So, do all of these dysregulations cause placental failure? So, as a proxy, because I think I only had like 2 months left in the US, so we did a simple experiment to show placental function. So the placenta, being an organ that not only functions as a barrier against foreign substances, it also is an organ that allows nutrition to come to the fetus from the mother. And so we used glucose transport as a proxy for placental function. One of the classical nutrients that goes through from the mother to the fetus is glucose. So we treated our cells with glucose. We added glucose, starting from the maternal circulation. We allowed glucose to flow downward to the fetal circulation. And then after 1 hour, we took the cells and then we fixed them. We fixed them. And then we see that cigarette smoke extract does not really induce a lot of glucose crossing. Because what should happen is that once we fix the cells, once you make them dead, of course, it's not going to function anymore. So glucose should flow freely from the maternal to the fetal circulation. But as you can see, even against control, all of these exposures are not sufficient to promote dysfunction, meaning that it would still allow for a normal flow of glucose from the mother to the baby. So in summary, low levels of pollutants are weak producers of massive placental dysfunction. That's why we don't see, right? We don't see a lot of patients having more critical phenotypes, such as fetal death, stillbirth, whenever they get exposed to these pollutants. But there is still an epidemiological basis for the negative effects of these pollutants. So what we are proposing is that these pollutants may only serve as weakening agents, meaning that it weakens the placenta, and once another insult comes along, it will then lead to a massive placental failure that may be leading to preterm birth. Not only that, epigenetic changes induced by these pollutants may also be a factor. And we are also proposing that because the placenta is a strong organ relatively, it rescues itself, as you can see. Maybe other organs, such as the fetal membranes, maybe the cervix, may contribute more to the preterm birth determining process. So in this study, we are able to show that we can do these drug testings, reproductive toxicology in just a small system. We are able to include human cell lines that replace traditional cancer cell lines. And it's a proof of concept also that we can include immune cells in our chip, therefore mimicking a more natural physiological state. And we can use this system in the future to test any other drug or any other compound of interest, because we can't really do toxicology studies with pregnant patients. That's going to be unethical. So what we are proposing is that for this system to be used in the future. Of course, we have some limitations. We acknowledge some limitations in our model. And more importantly, we need long-term pollutant exposure. As the lab still optimizes, maybe making these experiments not only across 3 days, maybe extended to probably a week, 2 weeks. It's still a matter of study. But for our purposes, we are able to show that we can create our own cell lines with higher physiological relevance. We are able to replicate the placenta function. We are able to demonstrate that low concentrations of these pollutants do not necessarily disrupt placental function. And finally, our cells and our chip can be used for future pregnancy units. So I would like to just acknowledge the panel members of my team. They guided me through a lot of different things, most especially my advisors, Dr. Yu and Dr. Menon. And most importantly, actually, that lab and the University of Texas Medical Branch at Galveston. It's a small lab, but we are able to do wonderful things for pregnant patients in that institution. Okay, and so yeah, it was a good journey in the U.S. I met a lot of people, made a lot of friends, and actually I've also met some of our doctors there in the U.S. Dr. Cabellion. I've seen him in Boston in July of 20, I think that was 23. So it was a good journey. And I would really like for everyone, like if there are people in the Philippines right now who wanted to do research, you can do it abroad and then just go back here and try to disseminate that research for everyone so that we can improve our own system here in the Philippines also. So that's it for my talk. Well, that was 30 minutes of your time. Thank you so much for your interest and your attention. And I'll be taking any questions from the audience.
Dr. Tanchenko, you have the floor to moderate the questions and answers.
All right. Thank you, Dr. Cabellion. Congratulations and thank you, Dr. Manuel Vidal. So Manuel is my batchmate and a really good friend. So as PAASE, I think this type of research is a good example on how collaborations among the basic sciences, medicine, and engineering can help us answer a lot of clinical questions. Because the devices were not developed by us clinicians and basic scientists. We developed it with the help of our friends from the biomedical engineering and the electrical engineering departments.
Yeah.
Thank you also. Yeah, so thank you also, Dr. Vidal, for studying placenta, which is, I think, personally one of the most neglected and understudied tissues in the body. despite it being involved in a lot of pregnancy complications like preeclampsia. And the lack of studies in placental biology led to, preeclampsia was discovered in the 1900s, and it's already 2024, and the only cure that we have is delivery, which is not foolproof because a lot of patients can still get preeclampsia and eclampsia after delivery. Also, thank you for enlightening us about bisphenols. You've shown several epidemiologic studies, but these are association studies. So we need more of these experimental studies, which will, try to establish causation between bisphenols and if this will really cause preeclampsia or preterm birth. And also, I attended one of the talks in the UP Marine Science Institute. They showed that microplastics are present in a lot of fish and aquaculture products that we eat. And bisphenols can actually adhere to these microplastics. So we are actually taking in a lot of these bisphenols probably, and other endocrine-disrupting chemicals. So thank you. And I will now open the floor to questions before I ask my own question. So do we have any questions from the floor? Dr. Gladys Completo.
Hi, good morning, everyone. I'm sorry I wasn't able to be there on time. So, because I'm traveling. Congratulations, Manuel. I don't know if you still remember me when you just graduated from LaSalle. Actually, I joined LaSalle. That was the time that I joined LaSalle. That's why I remember you. So I have a quick question. So are you able to repeat this, your on-chip organ model here in the Philippines? Do you plan to do that, and then if you're already starting, what are the hurdles, or is it easy to do it here?
Actually, again, Dr. Tentenko highlighted earlier that it's the main highlight in terms of working environment is the collaboration between the engineering side, which does all the chip work, and us clinicians and biology researchers. We do the cell side. So here, actually, that's why I joined LaSalle. I'm currently a faculty also of De La Salle University. Someone there is working with microphysiological systems. He is doing small systems of the liver and I think the brain also. So he's trying to do a small chip that also has different cell lines in a single system. So I started talks with him, and of course, I needed an institution. So I started also I joined also LaSalle, hoping that I can do work in their own lab. But I've already talked to them. And also, not only that, we just had our grant approved for the prepare program. So one project there is the establishment of Filipino preeclampsia cell lines. So the techniques that I've done in the US, we will be doing that also, so that we can also have our own cell lines that would be of more importance for preeclampsia studies, because Filipinos are naturally at higher risk for preeclampsia just because of our ethnicity. And so, since I don't think there is a cell line that is like a preeclampsia prone cell line, I don't think we have that one. So in our program, hopefully we can biobank and we can bank them and use them for our own future research.
Yeah, and then another follow-up. So it's good that you're already talking to the other researchers in De La Salle, and I think they will be able to help you. It's really amazing how you were able to show that you can, using your model, you can actually do immortalization of cell lines, right? Because in one of our projects that I was involved in, we tried to immortalize Filipino blank cancer cell line. It was from primary cells. It was really very difficult for us. We tried several. It was anyway. It took us a long time to do that. This was in partnership with UP Manila, so that's why I commend you for showing those results where you were able to show that you were able to, am I correct? Did I get it right? That you were able to immortalize it, right?
Yeah.
And yeah, and I hope, yes.
I'm sorry. I actually, before I came in, they were
Mhm.
trying to immortalize them, and they were having a hard time also.
Mhm.
And so they needed someone who will be working full time with that. And so I did a lot of optimizations. That was the longest part of my
Mhm.
my work. It took me like 1 year to immortalize properly that cell line. It's really hard, and I understand how hard it is for you also.
Yeah, so hopefully, yeah, we would like to contact you, and then maybe it would be good to
Yeah.
really do that as well on just on a microchip, the immortalization of because this is the first, we were the first lab that were able to do that, not my lab, but our collaborator as part of our project. So I hope we could collaborate and then maybe we will be able to be more successful in doing the other stages of lung cancer, because that was just only one stage that we were able to do. And I thank you very much. I think there's another question. Somebody's raising her hand, Dr. Victoria Herrero.
Yes, that was a great talk. Congratulations on your organ in a chip or placenta in a chip. Few questions. Have you validated what you've found with an in vivo model? Because as you know, there's always limitation of cultured cells. And for improvement, is this FDA accepted, for your future drug testing? At what stage are you in? And lastly, to improve your chip, would you consider co-culturing cells so that you could simulate cell-cell contact, which you do not have in your current system? It's all by flow. Because cell-cell contact and matrix cell contact will be very important, especially when you start getting into pathogenesis of preeclampsia. And one more question last. iPS cells perhaps is what you can develop from the Filipino preeclampsia patients, if there really is something very unique in the genetic makeup of the Filipino cells ethnicity.
So well, thank you for those questions. Well, the first question was, we have not yet that was the next step, because we acknowledge that of course this is an in vitro setup, so we would like to validate them. I think that they would do it side by side with preparing it for a larger scale, because this work was used to approve to ask for a grant, and it has been approved, to ask for a grant to do more work in the placenta on a chip. So I think they will be doing validation side by side with in vivo models. Probably it's going to be a mouse model also. So the next question was
Co-culturing or validating co-cultured cells with matrix.
Yeah. So in our well, maybe I can share my screen again here. If you see here, our chip actually has these channels. So I've not shown it here, but we were able to see that when you look at them in a microscope, cells from this chamber would go to this chamber also, and vice versa. So there is cell-to-cell contact, but it's limited by the number of channels. So eventually, what we would want is for it to come naturally as a natural contact. So removing these channels, making these chambers side by side together, so that they would be, be adjacent to one another and not limited by channels. There is also another parallel work with this one that uses 3D bioprinting. What they were doing is that they have a layer of cells, and then they have a layer of cells on top, and then they print them on a polymer. And so that would stimulate more that would more simulate a more physiological model. They have started doing work with that also. So we also have co-cultured one chamber here. This gray chamber here is for the supporting cells, the mesenchymal cells. We have introduced macrophages also in that area. And we were able to show that they do move across the chambers as what would you expect normally if you introduce an inflammatory insult. And so that's like an early level proof that we can co-culture these cells together in the same chamber. And we are hoping that we can add more immune cells in this one, NKs, T cells, not only here, but also in the decidua, which is the maternal tissue that is in contact with the fetal tissues. And
Well, actually, inflammatory cells should have contact with endothelial cells, because that's how you would have inflammation, endothelial interaction. And if you're looking at oxidative stress, your endothelial cells would also be activated. And the question is, does it activate the neutrophils or your other, inflammatory cells? And not to make it so complicated, you can do co-culture of endothelial cells, mesenchymal cells in the same chamber, that it isn't just, it's great that you're getting interaction and that they're attracting each other. And that's exactly the reason why you should be co-culturing it, because I bet you your results will be different once they're co-cultured for the cell-cell contact, not just flow, but contact. Anyway, yeah.
Well,
that's great. You've done great work, really.
Yeah, I mean, especially placental structure, but that's a really great question, and they are trying to answer it now by doing bioprinting, because they are able to layer cells more effectively without channels, without anything that would separate them greatly.
Yeah, and the last question was the iPS cells. If you're going to culture and immortalize, you should invest, if you're starting your lab in the Philippines, that's something that you can do there and will be unique for your Filipino.
It has been shown that you can convert a mature trophoblast, a mature placental cell, you can convert them back to their original state, because when egg and sperm meets, they fertilize, they develop into a blastocyst, and that cell is totipotent. Like it can differentiate into different cell lines. And they have shown recently, I think that was a research done in 22 or 23 maybe, that they are able to take a trophoblast, a mature trophoblast, treat them with chemicals, and they revert back to a state as if it was still a blastocyst. So because iPSCs are really expensive, I mean, it's expensive work. I think that would be more feasible if we had mature cells, and then we just treat them with chemicals, try to see if they go back to the same state. But nonetheless, iPSCs are a great way to differentiate them into different cell lines. And it would be cool if we have our own, like Filipino iPSCs, can develop them also. Because we are at risk for a lot of other diseases just because of our ethnicity. And so it would carry, iPSCs from Filipino populations would carry probably the same epigenetic, the same genetic predispositions that would be of more relevance to us Filipinos.
Thank you. So do you see a difference between preeclampsia and normal placenta? If you're going to get the primary cells, do you see a difference?
That is one of the projects that we have asked for a grant for and was recently approved. Dr. Tentenko led the team for that approval of that grant. So the second component of that project will be looking into Filipino cell lines, preeclampsia, and of course, comparing it with normal. But in the literature, they have seen that some of the genes, when it comes to vasculature and proliferation, they are dysregulated in preeclampsia patients compared to normal patients.
Great, thank you.
Thank you.
Thank you for your questions, Dr. Herrera and Dr. Completo. Yeah, we are actually excited about that project. We just recently received almost 100 million grant from the OSCP-CHRD to study preeclampsia in the Philippines, and hopefully we'll start around 2026, but it takes time for DOST to give us the money. But hopefully we can share that result. And we will also biobank the tissue. So definitely we cannot do iPSCs during this project, but it's not part of the objectives and also the LIB. But we have developed a method of cryopreserving the cells, so the tissues, so we can biobank the tissues and we can still isolate and establish cell lines from these biobank and cryopreserved tissues. So hopefully that's something that we can do after the current study. Also, for just want to comment on Dr. Completo with regards to the challenges in establishing an organ-on-chip lab here in the Philippines. I think the challenge is really with looking for the engineers, because when I came back to the Philippines, I also talked to the Triple E in UP Diliman, the electrical and electronics and communications engineering department. They don't have the expertise for it. So one of their instructors, Jean Manzano, I connected her to our collaborator in Texas A&M, and she's now doing her PhD in bioengineering there. And hopefully she returns in a few years' time so that I hope she returns so that we can do the work here in the Philippines.
If I could just add to that, because that engineering side, it's not just modeling something. You can create a lot of different, chamber designs. You can create anything. You can design anything. But to be optimal for cell survival, because I also tried designing, and I sent that one to my professor, and she said, "I'll think about it. I'll let my engineers look at it," and they said no. I mean, it wasn't really that functional. So I thought it was just an easy process of doing whatever design you like, but it's more of doing a design that would be functional for cell growth, attachment, and also communication.
Yeah, is it possible? Excuse me, is it possible if you let the engineers do it there in the States and then we can bring it back here?
Yeah, actually, one of my friends, she's in Brazil right now. Like, she was a student, Mary. She also took some of the, she created some of the chips. Then she asked for our principal investigators permission for her to bring some of the chips back to her country. And now they are trying to replicate the same technology. Of course, it's a collaboration because it would probably be a patent in a few years. And the important thing is probably just ask for permission if they would allow it.
Yeah, so just like a word of advice based on an experience, please don't give up. It's a little bit really very, very slow here in the Philippines. So don't get frustrated. Keep on still connect with your PI in the States, and then with the improvement of the procurement law, I think it will be faster now. So I'm very happy that you came back, Manuel, and also Arlad, and PAASE is here to help you. We've been mentoring a lot of young scientists. There's a lot of successful scientists in as PAASE members that are still giving back, although they are based in the States. they are willing to give back. Maybe we could connect you to some engineers that are based in the States as well, not just who your connection. So again, thank you very much, Manuel and Arlad. And I think Doc Billy has a question. I can tell.
I actually have a question, if I could ask. So these chips that you are creating, there are very specific properties, I guess, for in order for the cells to grow in there, not be contaminated, and all kinds of things that need to be done, plus matrigels where you can measure cell-cell communications without even touching them, and eventually cells touching each other. So in the future, is there, when they have perfected these kinds of, I know you created this for placental cells, can they be created so that you can test communication between cells, not just the placenta, but as well as applying it to other kinds of diseases, for instance? Like, for
Yeah.
instance, I'm a cancer researcher. We do microfluidics, and we have engineers that do microfluidics, and they're constantly bugging us for cells, But can these be in the future manufactured in bulk, like you would, for instance, buy tissue culture plates, something like that,
So the good
to
thing about
facilitate?
the design of some of their chips is that you can use it for different purposes. The primitive placenta model that they had consists of 3 chambers. So 2 of those chambers are for placental cells. One of that chamber is for the fetal vessels. So before I left, I helped them make a blood-brain barrier model using the same chip. What we did was we had one layer for a triple co-culture of microglia, astrocytes, and neurons, and then the second chamber was for pericytes. No, for blood vessels, and finally for pericytes. So not only are these chips specific, you can make these cells specific for your own function. You just have to know which cell types should be placed in them. And
Growth
also,
condition.
so you also have to consider, like, how do they look like? So what we did primitively was because we know in the blood-brain barrier that vessels are in really close contact with pericytes, so that you have a tight seal. So what we did was we cultured our cells vertically. Maybe imagine if this is your chip, these are the cells in here. What we did was we tilted them in such a way that the pericytes would fall down, creating like a kind of a tight seal. It was more of a primitive way to mimic the tightness of that blood-brain barrier. And I know in the Philippines right now, they are also, that group in De La Salle University have been working with liver chips, but they are, and you can, I think they have been repurposing that same chip design for the liver and for the brain.
So maybe it needs all, it just, you have to I guess I can imagine where you have the chip and then look at the growth conditions for your cells, plus, immune cells, and that's the one that you can change as you go to different kinds of systems. That would be a possibility, I think. Yeah, great. That's really awesome.
Dr. Vidal, I have one simple question. You said you immortalize these cells. How long do those cells in the chip live?
So earlier I've shown that we are not seeing that much of a cell death over 72 hours. So within 72 hours, the cell death within those chips tends to be around less than 10%, which is great because we know that they can still live for 2 days. We can still optimally do our experiment. But when we extend them to maybe 6, 7, we start to see that these cells detach. The media covering them starts to get murky, even though you keep them in sterile chambers. So the chip is dying within 7 days. So even though they are immortalized, it doesn't mean that they would be optimal to be outside their natural container for a few more days. So the challenge also would be to allow for these chips to be fully functional for more than a week, because not all exposures would just take 3 days, would only take 1 day. Not exposures are like that. For most of the exposures that we have in our body, in our system, we get exposed for months, especially in pregnancy. And so that's the next challenge, actually.
Thank you.
But the immortalized cells, you can take them out of the freezer every now and then. If you want to do experiments on them, you can just take them out of the freezer, culture them, and then just, passage them, just repeatedly passage them. And you can have our cells live for maybe 3 months on their own. So it's a kind of long process before they start dying.
Thank you,
That's
Dr.
still long enough
Cabellion.
to study a lot of things, I think. Yeah, because I know with my bone marrow cells, they live for about a week and then they're done, unless you put them back into the mouse. Yeah, that's really great.
Yeah.
Yeah.
I think most of our cell lines in the lab, they will survive 20 to 30 passages, and they can still grow. But I think the challenge in the chips, when they become too confluent, they start to die because the space is very limited. So
Because you probably cannot split them, right? Can you split them?
yeah,
No.
So that's the
no.
point. So this is why they die, I guess, after some time. Yeah.
Right. Do we have do we still have time for more questions?
I guess if people have
Maybe one or
questions,
two more? There any questions?
questions in the chat.
Yeah, any questions from our audience? If none, maybe I can ask in relation to immortalization, Dr. Vidal. You did transcriptomics and you compared your immortalized cell lines versus the commercially available vivo cell lines, other cell lines. But did you also compare your primary versus your immortalized cell lines, if there were changes in the transcriptomics, or if there were change? Because I know you compared the uh, NF-kappa B expression in the P38, but with regards to the transcriptome, did it change when you immortalized it versus the primary cell?
So when we took the primary cells from the placenta, it's hard to maintain them in culture unless you immortalize them. So once they are immortalized, the primary cells are good for like 3 months. They're good. But for primary cells, the challenge is to maintain them alive within 1 week. And so they tend to grow really slow when they are in their primary state. I've done a lot of different stimulants just to keep them alive. So it was a challenge for us to get enough quantity for transcriptomics. I think there was only one successful time when we were able to do Western blots for like the CSE exposure, for the LPS exposure, but that was only one time. And we only had one flask. That is only for one experiment. So we were not able to grow them again in the optimal conditions, enough for us to get the right amount for transcriptomics. But we had one sample, I think, but that's not a replicate. We don't have replicates for it. We had one sample. But the overall transcriptomic signature for that single primary placental cell was in cluster with the immortalized placenta cell that we had. And it was way off from the also way off from the BO cell. So we thought that, maybe we can show this data to say that they're not really significant. Our immortalized cells with an N of 3 are not significantly different from the primary cells. But that was just an N of 1 for the primary cells. So we didn't include that in the paper about that.
I see. Because if you think about it, maybe other researchers will say that, oh, this is like a cancer cell line, because when we
Right.
immortalize it, we treat
Right.
them with SB40, EBV,
Right.
you know, so it
It no
might be
longer
good
reflects
to,
what's normal.
yeah,
Yeah.
so it might be also be good if you can show that there's not much difference in terms of the transcriptome. So
But we
because
are able to show that, but just with an N
I
of
see.
one.
Because there's this one immunofluorescence data that you showed that the vimentin staining primary versus the immortalized, I think it decreased significantly. I can no longer see vimentin staining. So is there a change in mesenchymal to epithelial transition? Do they lose their mesenchymal properties when you immortalize them? So
Oh, wait. Here.
yeah, so you can see that the vimentin staining significantly decreased when you immortalized it. So
Yeah.
um,
When this is to show that in primary cultures of the placenta, you get a mixture of the placenta cells and the mesenchymal cells. So the placenta cells are not supposed,
oh,
the placenta cells are not supposed to express vimentin. So we showed that here in the primary cultures that we had, we get contamination of mesenchymals. Once we immortalize them, it overgrows that other mesenchymal cell, those mesenchymal cells, and we no longer see vimentin stain, which is good because we know that our culture is now primarily placenta and not mesenchymal.
I see. So yeah, I didn't get that uh, in your presentation. So the immortalization only targets the trophoblast and it discriminates against these mesenchymal stromal cell lines,
Yes.
correct? Okay. All right. But these stromal cell lines, did you use the same immortalization technique for the trophoblast and the stromal cell line?
Interestingly, stromal cells, they're really proliferative. We take them out, primary culture of them, you can, they stay alive for 30, 40 passages. Because if you're familiar, if everyone's familiar with regenerative cells from the placenta, right? They sometimes use cells from the placenta for regenerative purposes, especially for plastic surgery, so that they regenerate the skin after burns. So they use the placenta, the mesenchymal cells in the placenta. So there's no need for us to immortalize them because they stay alive. They are highly regenerative.
Great.
Interesting.
Thank you so much. Do we have more questions? Any more questions from the attendees?
I just want to say that you have a very fascinating system and really good luck with the research in the Philippines and looking forward to more of your results.
Yeah, we hope to do this research here in the Philippines. Again, we need engineers. engineers. We
have
need
engineers in the audience.
we need to immortalize cells also, but that is, I mean, the grant that we are working, the grant that we are going to work for already will cover isolation of gestational cells from Filipinos. We just need an engineer for us to do.
Yeah. Hopefully we can get collaborators from the engineering side because it's really difficult to understand fluid dynamics. They do a lot of computations about this that my brain cells cannot process.
Yeah, understand.
So maybe the electrical engineering department in Utilos Baños they're really actually very good also. So maybe you would like to consider contacting one of them. So anyways, yes, PAASE will help you, and then we could look for DLSU has a very good engineering department as well. So I think you're in the right university right now, Manuel. So
I just need to be full-time faculty there for me to start research.
yes, you should. Okay, so okay. So
We'll see.
Please, but again, as I've mentioned, please don't give up. I hope you do stay here in the Philippines and you can start your work here in LaSalle. And go talk to Raymond Tan, okay? I'm sure he he'll do. Raymondton is the right for the research, vice president for research in LaSalle. Yeah,
Have you met Raymond?
You should
We should
talk to
introduce
him.
him to Raymond.
Yes,
Oh,
I will
my
introduce.
friend.
Yeah.
Angeline is here. Where's Angeli?
Angeline. So yeah, I'm sure he'll take care of you, Manuel. So just go talk to him. And LaSalle also has a separate funding, right, for new faculty members. So congratulations for getting that funding from DOST. Is it Fishered or PCHRD? So congratulations. When will it be? When will it start? Will it be 2 years from now, or will it be next year? Your
26.
grant.
2026.
Okay.
Yes.
Okay. So yeah. So while you're waiting for that, hopefully LaSalle can provide funding for you so that you can start like a seed grant. Okay. So they can do that.
When we had our cancer center, we had some
Breaking.
markers were. I will try to find them, and if I find them, I will invite them to collaborate with you because they are very, very passionate about all these things. Yeah. Okay. So if we have no other questions, Gladys, I guess you're giving the closing remarks now since I started the whole thing.
Okay, but before we do that, can we give the certificate to Manuel? Fab, do you have the certificate? Yes. Okay, so again, thank you very much. We would like to present the certificate of appreciation to Manuel Vidal Jr., medical doctor, for being our speaker at the Filipino-American Academy of Science and Engineering, or PAASE webinar entitled The Development of a Placenta-on-a-Chip Model to Evaluate the Effects of Endocrine-Disrupting Compounds in Inducing Acute Placental Dysfunction, held on October 18, 2024, presented this day, October 18, 2024, with our deepest gratitude, signed by yours truly. Again, thank you very much for this exciting research that you have shared with us. And we also have other, this is just the first leg. We're actually celebrating the Filipino-American History Month this month. So we have other webinars that we'll be hosting. So this will be on October 25. The seminar will be presented by Maria Zinia Agustin, a professor at the Department of Mathematics and Statistics from Southern Illinois University, Edwardsville. And the title of her talk will be Enrichment Sessions in Calculus 1, a Path to Retaining and Graduating Engineering Majors. So that's on October 25, the second leg for this webinar series. And on October 28th, this will be the last webinar for this Filipino-American History Month celebration. This will be face-to-face to be presented by Gobert Advincula from the University of Tennessee, Knoxville. He'll be here as a balik scientist. He'll be back in the Philippines starting October 25th. He'll be staying until October 30th. And he'll be going to University of the Philippines, Los Baños, to talk about polymer materials, AI, artificial intelligence, and machine learning and advanced manufacturing for Philippine innovationeering. Okay, so those are other webinars. That's all for, oh yes, this is for November. Okay, in November, Christian Malapit of Department of Chemistry, Northwestern University, will also be giving a talk. Okay, so, and Dr. Neil Concibito from Osaka, Japan, Corny Incorporated, will be also giving a talk on promoting process safety management in Philippine universities. Actually also a balik scientist. He was in UP Los Baños last year to give his, to serve as a balik scientist. Okay, and
We have
Lastly, another one, Dr. Aristotle Obando. He'll be giving a talk on application of PAASE theory on the sustainable production of microalgal biofuels. So this will be on December. So these are all the webinars that are scheduled for this month until December. Please do, if you have time, please do register and attend our webinars. So as closing, I would like again to thank everyone for joining us today for this exciting session as we celebrate the Filipino-American History Month. It is just very fitting time to highlight research done by our young scientists here, just like Manuel. And we would like to invite Arlad as well to present your work. If you have the time, please do present your research work. And because all of your research highlights and contributes to the global scientific community. So Manuel gave us a very good talk. His study on that explores the impact of endocrine disrupting compounds, especially bisphenols, on placental health, using an innovative placenta-on-a-chip model, and hopefully he will be able to replicate this here in the Philippines. This research has the potential to shed new light on how these compounds contribute to critical conditions like preterm birth. So the implications of this study could revolutionize our understanding of maternal and fetal health, providing us with valuable insights into preventing adverse pregnancy outcomes. So again, thank you very much for your presentation, Manuel, and it was a very good discussion. It will truly inspire new ideas and collaborations in this area of research. And by the way, this is being recorded, so people can still watch your presentation, Manuel. You can just send them the link, and it's available to our FB, in our YouTube channel as well. So please feel free to share. We will also give you the recording so that you can share it to your students. Are you also teaching now, Manuel? You can share that. So again, thank you very much for your wonderful presentation, and I'd like to thank also Dr. Billy Cabellion for the initiative, for inviting Dr. Manuel Vidal, and for Dr. Arla Dantenko for being our moderator. We look forward to you being part of PAASE. So I hope you will be involved in all of our activities, and we will look for engineers that could help you start your research here in the Philippines. Maraming salamat po for all the attendees this morning. Thank you.
Thank you po for
Can we have a picture, Fed?
people have to show their faces.
Okay.
Turn on your cameras po.
Turn on your cameras.
It's just showing my face here. As can we have it in panel?
Yes po. Okay po. 1, 2, 3. Smile. Yan. Isa pa po. 1, 2, 3. Smile. Okay na po. Thank you.
Thank you very much again. Thank you so much.
Thank you very much.
Thank you,
See
Pat.
you next week.
Thank you,
Next
Dr.
week.
Cabello.
See you next week. Next week.
Next week. Okay, yeah.
Yes.
Next week, another one.
Okay.
Okay.
Bye.
Bye. Good morning.
Good morning. Good night, Marge.
Oh,
Good
gabi na
night,
rin.
Billy. Okay, bye.